Hi, my name is Jeff Hoffman, and in this lecture I'll cover the lab techniques that are useful to know for the step 1. The polymerase chain reaction, or PCR, is used to amplify DNA. This just means making copies of DNA from what you already have, and you're only amplifying a specific piece of DNA that's maybe a few hundred base pairs long. So why would you want to do this? Well, first of all, if you want to see if a particular sequence of DNA is present in a sample, so for example if you suspect a patient has a viral infection, you could use PCR to try to amplify a DNA sequence that's specific to that virus, and if nothing is amplified, then that virus probably wasn't present in your sample. It's also useful if you want to take a closer look at the sequence of a particular site in the patient's genome. For example, if you want to look for a mutation or a version of a gene that might cause a disease. Okay, so how do we do PCR? Well, to start with, you need your DNA sample called the template, which is shown here, and you also need a heat-stable DNA polymerase, which is usually Taq polymerase, from the thermophilic bacterium Thermus aquaticus. You need nucleotide primers, which are complementary to the ends of your DNA template, and of course, the 4 types of nucleotide triphosphates, which are the building blocks for your new DNA strands. These are all combined together and put in a machine called a thermal cycler, which does the rest of the work for you. A thermal cycler, as you might guess from its name, cycles between high and low temperatures. The first step is to heat the DNA, which melts it, to allow the strands to separate from each other. Next, it cycles back to a lower temperature, which allows the primers to bind to the single strands of DNA. One primer is complementary to each strand of DNA. The only reason the primers bind to the template rather than the 2 template strands binding to each other is because the primers are much more abundant. The final step is DNA polymerase making a new strand of DNA, starting from the primer and using the original strand as a template. This results in 2 pieces of double-stranded DNA, which is twice as much as you had before, at least for that particular region. By repeating these steps a bunch of times, which is as easy as letting the temperature increase and decrease over and over again, you can end up with many orders of magnitude more copies of DNA than what you started with, because you double it every time. For example, 30 cycles would give you 1 billion times more DNA than what you started with, because 2 to the 30th is about 1 billion. Next, you'll want to make sure you amplify the right thing, which you can do if you know how long the DNA you're trying to amplify is. You can do this by running the PCR products through an agarose gel using electrophoresis, which will separate the fragments by size. Since you now have billions of copies of this particular small region of DNA, it's the only thing you're going to see. By comparing it to this DNA ladder, which contains fragments of known sizes, you can determine whether you amplify the right piece of DNA. Electrophoresis of DNA through a gel is also used in our next lab technique, the Southern blot, which you can see an image of here. This is named for its inventor, Edwin Southern, and the names of the other types of blots we'll talk about, Northern and Western blots, are kind of playing off this name. The purpose of a Southern blot is to detect specific sequences of DNA, which can help you diagnose genetic disorders or the presence of particular viruses or bacteria, or even be used for forensic applications, such as whether a drop of blood came from a certain person. First, you have to load your DNA of interest into a well at this end of the gel. Next, you apply an electric current to the gel with a negative charge at the top end and a positive charge at the far end. Do you know why you align the charges this way? It's because DNA is negatively charged, so it will run away from the negative end towards the positive charge. Since the gel is basically a tightly woven sponge of fibers, larger pieces of DNA take a long time to get through the gel, whereas small pieces get through pretty quickly. This principle is what allows you to separate strands of DNA by size. When you've been running this long enough that the smallest strands of DNA are nearing the end of the gel, you shut off the electric current and transfer the DNA to a filter, where the DNA is more stable and can't diffuse around. At this point, you can't see what's on the filter, but you can use a radio-labeled DNA probe that is complementary to, and therefore will stick to, whatever sequence you're interested in. So for example, let's say you suspect your patient has a particular viral infection, and you know that virus has a certain sequence of DNA that is not found in humans. You can create an oligonucleotide that is complementary to that sequence and attach a radioactive label to it so you can see it later, and then wash it over your filter. After you give it some time to stick to its target, you wash it away, so the only probe left is in the location of its complementary target sequence. If the target sequence that you're looking for was not in your original sample, then the labeled probe will all wash off and you won't see anything. But if it was in your sample, then it will stick to its complementary sequence and will show up as a band on the filter on the X-ray film. This is the only thing you'll see, since only your probe is radio-labeled, so the rest of the DNA on the filter won't be visible. A Northern blot is basically the same thing, except now instead of running DNA through your gel, you're looking at RNA. So this can be used to measure the quantity of RNA, or just whether a certain sequence of RNA is present. This is often used to determine how much a certain gene is being transcribed into mRNA. A Western blot also uses a similar concept, except now you're looking at proteins. Since proteins can't be targeted by using complementary strands, you instead have to use labeled antibodies which are specific to your protein of interest. One example of when Western blots are used is to confirm the diagnosis of HIV after it has been detected using an ELISA, which we'll discuss shortly. Western blots can be used in any case when you want to know how much of a certain protein you have in your sample. And lastly, a southwestern blot is kind of a hybrid between the southern and western blot, so you can use its name to remember this. The goal here is to detect proteins that can bind to a specific sequence of DNA, such as transcription factors and nucleases and histones. The way you do this is to run a protein through a gel just like you would during a western blot, but then use labeled oligonucleotides as probes instead of antibodies. If the DNA probes stick to a protein, you will see a signal at the location in the gel which corresponds to proteins of a certain molecular weight, and that lets you know which proteins can bind to that sequence of DNA. A microarray is a very powerful lab technique since it allows you to perform thousands of genetic tests at the same time. The most common purpose of microarrays is to look at the level of expression of all genes in the genome. The microarray itself is a small chip about the size of a penny that has thousands of DNA or RNA probes attached to it. Each probe is usually about 25 base pairs long, and is complementary to part of one specific gene, and they're arranged in a specific predetermined pattern on the array. The way you use a microarray is first you take your tissue or cells of interest, isolate RNA from it, and then you can label that RNA with a fluorescent tag. Then you can spread your RNA across the chip, and after letting it sit for a while, you wash it off. Any RNA that was complementary to the probes in the chip will stay there. So if a particular gene is expressed at a high level, the RNA from that gene will be more abundant, and more probes in the chip that are complementary to the part of that gene will bind to its corresponding RNA. Next, you let a machine read the chip by looking for those fluorescent labels, and since the probes are arranged in a specific order, the machine knows which ones correspond to which genes. Microarrays are particularly useful when you want to look at a large number of genes, such as when looking for predisposition to diseases or mutations that may have given rise to a tumor. They can also be used to detect single nucleotide polymorphisms, or SNPs, which can be used for genotyping or linkage analysis. Let's quickly review with a flash quiz. A southern blot is used to visualize blank, a northern blot is for blank, and a western blot is for blank. So this would be DNA, RNA, and protein. By the way, I like to remember this using the top half of a picture of a cell. So here's our nucleus at the bottom, and here's our plasma membrane at the top. The DNA is in the nucleus, so this is the southern region for southern blot, and then it gets transcribed, and the RNA is in the north for northern blot, and the RNA gets translated, which I'm going to draw in the west, so the protein is in the west for western blot. If you don't like this, then don't use it, but hopefully it helps some people. Our next lab protocol is called ELISA, which stands for Enzyme-Linked Immunosorbent Assay. As you might guess from its name, ELISA is an immunologic technique, which uses specific antibodies or antigens to detect the presence of a target that you're interested in. There are two types of ELISA, direct and indirect, and both are typically done on a microtitered plate like this one, and you can test a different sample in each well of the plate. For a direct ELISA, we want to see if an antigen is present in a sample, for example in our blood or serum. So let's say we want to know if and how much of an antigen represented by this orange star is present in our serum. First, we put the whole sample into a well in our plate, and then fix the antigens to the bottom of the well. Then we add some antibodies that are specific to our antigen of interest, which again is the orange star. We want enough antibodies to make sure to hit all of our target antigen that's present if we want this assay to be quantitative. Then we wash the plate, and all of the antibody that isn't bound to an orange star is washed away. Since this antibody is linked to an enzyme that can generate a color, we simply add the substrate for that enzyme and it will generate a colored product. So if our antigen wasn't present in the original sample, we wouldn't see any blue color here. And if there's a little bit, we'd see a little bit of color, and if there's a lot, we'll see a lot of color. We can then quantify how much of color there is to figure out how much our original antigen was present in the sample. For an indirect ELISA, we can either look for an antigen or an antibody. Let's start by looking for the same orange star as before. The first few steps are the same, but there's just one more step here. In this case, our primary antibody doesn't have an enzyme attached to it. Instead, we can now add a secondary antibody, which is specific for the Fc part of our primary antibody, and then wash away the excess. The secondary antibody has the color-generating enzyme, and we can use it to generate a color and quantify the original amount of antigen just like before. So why would we do indirect when we could save ourselves a step by doing a direct ELISA? It's mostly a matter of convenience because it's easier to have a stock of labeled antibodies that target the Fc portion of a wide variety of other antibodies, like these secondary ones, rather than have to put a label on each primary antibody you'll ever use. Okay, one last thing. So how do we determine how much antibody is present in a sample? Let's say we want to know if this orange antibody is in a sample. We have to already know what its target is, which in this case will be the black star, and we have to start by having a well with that target antigen fixed to it. Then, kind of like before, we add our sample, which of course is a variety of antibodies, but when we wash it all off the plate, only our antibody of interest will be left behind. Now, just like before, we can add a secondary antibody which targets the Fc portion of our antibody of interest, which is linked to a color-generating enzyme, and by measuring the color we can infer if and how much of our orange antibody was in the original sample. So when you're done, you might end up with a plate that looks like this, if the sample is towards the bottom of a higher concentration of the antigen or antibody you're testing for. And the plate can be read by a plate reader to tell you exactly how much signal you're seeing. ELISA tests are extremely sensitive, so they're often used to screen for a disease such as HIV. Since false positives are somewhat common, these tests can be confirmed by a more specific test, such as a Western blot. So Western blots and ELISA both use antibodies to detect a target antigen. But can you think of why a Western blot might have better specificity? One reason is that the Western blot allows you to observe the size of what the antibodies are sticking to. So if they stick to something that's the wrong size, you'll realize the test is a false positive, whereas with ELISA, all you know is that it's stuck to something, but you have no other information about what you're detecting. The last technique we'll cover is the karyotype. Karyotyping is the process of staining, organizing, and numbering chromosomes based on morphology, size, and ratio of arm lengths. This can only be done when DNA is condensed, which is at metaphase, so usually you would use a drug that halts the cell cycle specifically at metaphase first. Can you think of a drug that's commonly used for this? Usually you'd use colchicine. You end up with an image like this one, which allows you to see all the chromosomes so you can look for gross abnormalities and diagnose diseases such as chromosome imbalances. For example, patients with Down syndrome would have 3 copies of chromosome 21. Fluorescence in situ hybridization, or FISH, allows you to directly visualize chromosomal anomalies that are too small to be seen in a karyotype. You start by preparing either a tissue section or cells on a slide, and then add DNA probes that are complementary to the sequences that you're interested in. Most cells will probably be in interphase, so they'll look like this one on the bottom, since the chromosomes aren't condensed, but cells in metaphase, which you'll be more likely to see in rapidly dividing cells or in cell culture, will look like this example on top, which can be more informative. Since you can label the probes with different colors, you can detect multiple sequences at the same time. You can even use lots of different colored probes to do what's called chromosome painting, to make a chromosome look like a rainbow in which each color corresponds to a different region. Then, you'll put your sample under a fluorescence microscope that can detect specific wavelengths of light. If the sequence you're looking for is present, then you will see the color or wavelength of light that corresponds to the oligonucleotide probe that's complementary to it. So here we have a green probe and a red probe that are corresponding to 2 different regions of the genome. If the sequence is not present, or is mutated in such a way that prevents the probe from binding to it, then you won't see a fluorescent signal, and you'll know that that sequence is not in your sample. Normally you expect to see 2 copies of each probe, 1 for each chromosome. But if 1 chromosome has a deletion, then you'll only see 1 signal. So in this example we're using FISH to look for the 22q11 locus, and we see that this patient only has 1. So what did we just diagnose, and what symptoms would you expect this patient to have? Well, this could be DeGeorge syndrome or velocardiofacial syndrome, and this patient might have a cleft palate, abnormal facies, thymic aplasia, cardiac defects, and hypocalcemia due to parathyroid aplasia. The mnemonic you can use to remember this is CASH22, but I'll talk about this more in the genetics section. By the way, FISH is the main way you detect this, and affected patients are heterozygous, so they have 1 present copy and 1 deletion.